Great. Nice to have you here, everybody. Hi, this is Hossein from AZAR Innovations. Nice to have you here. Excited to have this webinar together. So this webinar is, we call it Organ-on-a-Chip Meetup. It's a meetup for us to show you what we are going to do in Organ-on-a-Chip Masterclass in London between 7 and 8 March this year. And yes, so this is going to happen 7 and 8 March in London. As I said, there's still some seats left, so please be quick and register before we start with a few online modules before the masterclass. Okay, something about the masterclasses I would like to mention. In the past 2 years, we have been doing the masterclasses. We are based in the Netherlands. We started in the Netherlands and then expanded to Europe. To other cities in Europe, and recently in India. Of course, we have this masterclass in the UK in March, but this year still we have more masterclasses in the Netherlands, but also in Turkey and Saudi Arabia, and we go wherever the demands. Who are we? So we are AZAR Innovations. We are basically scientists helping other scientists with adoption and use of organ-on-chip. We have no bias on the technology. We try to be independent, and we try to help you understand the advantages and disadvantages of the models, the available models, and adopt the technology that is closest to your applications or idea. And we try to be a bridge between, of course, biological ideas and engineering solutions. And for that, that's why we organize these organ-on-chip masterclasses and trainings. And we try to help you understand these technologies and connect you to the right technologies, specifically commercial technologies. And if and only if there is no technology out there that fits your model, we can team up with you to do R&D for your idea. So something about the masterclass we're going to have in the UK. We start with an online module, which is e-learning 2 weeks before the masterclass to get you prepared, give you the information that is important to you for the in-person training. And then during the in-person training, we have 5 different modules that I'm going to talk about. We have 2 days, of course. Day 1, we're going to start with interactive lectures. We call them Learn and Design. It's a combination of lectures and, for example, tools where you can use to design your own system. And that's, you have heard correctly. These 2 days, you're going to design your own system. You're divided into groups. You're going to have to network, chat with your peers in the group, and at the end, you're going to design a system based on the available technologies that we have introduced to you. And on the first day, we also have a hands-on lab session. This is really the first experience for you to really handle chips, handle perfusion systems, and really understand what's going on with these systems. And then day 2, we have nice commercial technologies that you're going to see today as well. And we have 2 experts, and at the end, you're going to pitch your idea, and that's the moment that you're going to receive a lot of valuable feedback about the system you have chosen or designed. We have 2 great experts. I'm really happy about experts that we are having, Dr. Clive Roper and Pelling Chandarlioglu, both experienced people in the field of microphysiological systems and organ-on-a-chip. With industry experience. They are going to give a talk to you about available technologies, the standards, and more. And the cool thing is that they are there to coach you designing your own system. So it's going to be very fruitful discussion with them as well during the training. And of course, we have 4 organ-on-chip companies. I'm also happy because they come from different, they look at organ-on-chip from different angles, and they can help you as well with showing you their products, but also giving you insights on your design as well. Before I end the presentation, my presentation, I just want to mention that there's a workshop about immunocompetent models and lymph nodes, MPS. This workshop is organized between Triage Collaborative and IQ MPS, and it's a presentation of commercial products that offer these models. And this is part of a workshop series from Triage Collaborative and IQ MPS. All the important links are in the chat. You can go to the chat and see the links. If you want to see more of the masterclasses and also register for the masterclass in the UK, you can go to the chat and see the links there as well. And at the end, just the plan for today. So after me, Dr. Stephanie Moody from Animal Free Research UK is going to present their institution. This masterclass is co-organized with the nice people at Animal Free Research UK. We are enjoying our collaboration with them to make this happen in London. And afterwards, we are going to have presentations with from ibidi, Microfluidic ChipShop, Netri, and InSphero. And then we're going to close around 3:15 Central European time. So that was my part. I'd like to ask Dr. Modi to start sharing her slides and let us know more about Animal Free Research UK. Floor is yours, Stephanie.
Thank you very much, Hossein. I will just share my slides. All right, hopefully you should be able to see that. Let me know if you can't. Great.
It's okay. Yeah.
Thank you. Yeah, so as Hossein mentioned, we're working in collaboration with AZAR Innovation to bring about this workshop in London, which we're really excited about. So I'm going to do a very short presentation before our main speakers, a little bit about the charity. So as Hossein mentioned, my name is Dr. Stephanie Modi, Science Manager at Animal Free Research UK. And our goal at the charity is to support scientists to transition to non-animal technologies, otherwise known as new approach methodologies. And this is for the benefit of biomedical science to bring better treatments to people sooner through human-focused research, and also from the ethical perspective to replace animals in research, and also in the pursuit of scientific excellence through this human-focused research and the increased translatability to people. And we take a very practical approach to this. We understand that scientists can't transition overnight, so we do what we can to support scientists on this transition in a step-by-step way. So this next slide, I want to give you a little snapshot, a non-exhaustive snapshot of just some of the things that we do at the charity. So we fund research, we fund mainly pilot and transition grants. So pilot grants are sort of proof of concept grants that allow researchers to then apply for larger, longer grants, and also transition grants, so that scientists can transition from using animal methods or animal-derived biomaterials to fully humanized grants. And we've been running since about 1970, and through that time have funded over 260 grants. A key part of our support is to early career scientists. So we fund a number of PhD students. For example, we have 2 animal replacement centres of excellence in Exeter and in London, where we fund a number of PhDs. And obviously this training programme, we offer subsidies to early career researchers. So we're trying to support that way as well. We also run events. So we had our science conference last October, and we brought researchers together to help transition to human relevant research methods. And we have our online community of practice. So this is our online community where you can join up and network with like-minded scientists, hear about news, events, funding opportunities, and also join forums where you can talk and share expertise with other scientists. Fundraising is a big part of the charity, so we want to inspire the public to support the work we do. We do a lot of science translation and communications, blogs and news, things like that, to engage the public. And we run a number of projects. So one example of one of our science projects currently running is we are looking to help scientists transition to animal-free antibodies and find those animal-free antibodies more easily. So we're developing a recombinant antibodies database, which will be launched hopefully later in the year. And whilst we used to very much focus on academia, a new part of our strategy is not only to focus on academia, but also build relationships with pharma and biotech as well. We have lots of resources as well on our website. So we have something called the NAMS interface, where we've got on our community of practice lots of companies. Currently got 167 companies listed, so you can find companies that are using NAMS and developing NAMS, and also 34 animal-free biomaterial providers and 53 resources, including lots of non-animal method databases, for example. And we're active in policy work as well. So we present and do a lot of publishing, and we also drive policy changes as well by engaging with the government. So, for example, I did some Home Office training yesterday for the new licence inspectors, so that they can be more aware of what resources are out there when they are granting licences for animal experiments. On our website, as you can see on the right-hand side, we have lots of lay summaries of all our research, current research and past research as well, if you want to read about some of the research that we're doing. Little snapshot there of the various types of research. It spans quite a vast spectrum. So microfluidic mini brain research, animal-derived biomaterial replacement research, for example, gelatin. Humanization projects involving microfluidics and 3D brain organoids, new generations of muscle organoids, also projects that have been involved using 3D cell scaffolds made from plants, 3D models of blood-brain barriers, and an animal-free COVID-19 test. Also human stem cell models to study cardiac fibrosis. And also sort of some biomedical technology studies. So one of our researchers is developing a sensor to rapidly diagnose opioid overdoses. So a very wide spectrum. Going forward, we're going to have a more focused approach and sort of do grant calls on specific themes. So the next one will be around drug-induced DILI, which is drug-induced liver injury, for example. So it will be a bit more focused going forwards. This is just a quick snapshot of our NAMS technology hub. So this is the NAMS interface that I mentioned on the community of practice. So you can see at a glance lots of companies that are offering those NAMS technologies, and also lots of resources out there to help researchers find what they need. And very quick snapshot of our recombinant antibodies database. So this is a database that's going to offer really well-defined, clear and transparent information on the origin and production of recombinant antibodies, including the use of animal-derived biomaterials. And our antibodies will be categorized into 9 different categories depending on this, and you can filter according to what you're searching for in terms of the production, but also the application, like ELISA, Western blot, etc. There's also the opportunity to find completely animal-free antibodies, which are the green categories. And also you can find suppliers that can custom make antibodies for you. So that's a huge shift away from the animal antibodies produced in animals, for example, the polyclonals, which are still quite widely used. And this is our quick glance at some of the publications, quite wide-ranging, from Parkinson's through to proceedings from workshops and publications on modernising medical research using NAMS. So charity is very much a thought leader in sort of pushing forward that transition towards a new approach methodologies. And if you want to read more, you can look at our news and blogs, lots of different examples on there, like our Helpathon, which was an innovative event to bring people together. Some of the animal-derived biomaterials replacement research that's going on, and cancer technologies, and things like policy as well, like the FDA Modernisation Act 2.0, which was a bill that was passed in the US, allowing some in vitro testing to replace certain parts of the drug development process. So really wide-ranging articles on there. And we also have a podcast as well that you can listen to. So there's Dr. Davina Samose, who was one of the guests. So some really interesting life stories from scientists on that podcast that you can listen to as well. And lots of resources for researchers. So obviously you can see our Organ on a Chip masterclass there in relation to this masterclass. Information about funding and our conference, our community of practice, helpathon and grant holder papers, that sort of thing. So that's a very quick tour of our charity. Hope you find that useful, and I'm happy for you to get in touch as well after this presentation if you have any additional questions. So thank you very much for your attention.
Thank you very much, Stephanie. I specifically like your community where you post different resources about different, at least different models that I'm interested in. So if you have questions about the masterclass or have questions about the activities of Animal-Free Research UK, you can write them in the Q&A and we're going to answer them at the end. But we're going to start with the commercial presentations with Dr. Peggy Banish from ibidi. Nice that you're here, Peggy. Floor is yours.
Thank you very much. Thanks for the nice introduction. So yeah, I'm Peggy. I'm from ibidi, and today I would like to give you more information why it's important to choose the right microscopy chamber when you want to image your 3D cell aggregates, for example, and in combination with our solution for perfusion of spheroids. Starting with this, what is ibidi? Well, ibidi is a company whose goal it is to make you successful in cell microscopy and cell-based assays. And the images that you've seen here or see here have been done by our users worldwide, and they are showcased in our calendar for this year. So very beautiful. And any one of these people used any kind of these different kinds of labware that we offer. You can see this is a whole range of very simple ones like the regular dishes, plates, 8 wells. But we also have very unique designs like channel slides for flow applications. We have solutions for 3D cell culture, which is the topic today, and quite unique designs to study certain assays like chemotaxis, angiogenesis, and so on. And all of these have something in common. They are perfectly suitable for microscopy because labware has to fill certain parameters to make you successful. Not only is the microscope important because it doesn't matter how sophisticated it is. If you don't use the right labware with the perfect optics, you wouldn't get such beautiful results as I showcase here. Again, these are images done by our users from all around the world. And here I focus on organoids, spheroids, and beautiful images. And it's important to get them to also have a look at the microscopy chamber you use. And speaking of, we have lots of microscopy chambers. And just as an example, I choose the microslide 8 well high here. All our labware comes with a cover slip 1.5 at the bottom. They are suitable, therefore, for inverted microscopes. And we have 2 versions. We have polymer cover slip version or a glass cover slip version. So it's up to you what you choose. And both versions fulfill the following requirement. A number 1.5 cover slip always refers to the thickness of the cover slip. So there are different numbers, a whole nomenclature, but 1.5 refers to 170 microns plus minus 10. And most objective lenses are optimized for that. So no matter what kind of microscope you use, most objective lenses are optimized for this thickness to make sure that when you do high resolution imaging, that the focus of the objective is ending up where the cells are sitting or where your 3D silk aggregates are sitting. A negative example, a very exaggerated one, but I think it makes my point. When you use the wrong microscopy chamber, maybe with a very thick bottom, the focus when doing high resolution imaging with high magnifying objective lenses will end up inside of the material instead of the probe. Simple as that. And our labware solutions, of course, come with a 1.5 cover slip. And this cover slip fulfills other requirements. It's always important that you use imaging chambers that have, or the material itself has very low autofluorescence, because when you do fluorescence imaging, you will otherwise have a very strong background, as you can see in this negative example here. And yeah, no beautiful images with that. Another requirement for good optical properties is a broad range of transmission. So transmission of light should cover a broad range of wavelengths. So best between UV, visible light, and infrared. And last but not least, there are other important parameters I'm not going into detail on, but refractive index, it has to have a certain number, 1.52, around that. And an Ebbe number more or equal to 55. And our ibidi chambers fulfill these parameters, no matter if it's a glass cover slip or polymer cover slip. So the question arises when to use what, right? Our recommendation is as follows. If you do super resolution microscopy or TÜRF, we always recommend go with glass. Our tools come with a glass cover slip 1.5H at the bottom even. That's the highest quality you can get. It refers to less tolerances in the thickness. So it's only ±5 instead of ±10 microns. So the super resolution, go with glass, no question. But the polymer cover slip tools are suitable for the everyday microscopy applications like wide field microscopy, phase contrast, of course, confocal, even multifocal microscopy works perfectly fine. They have almost the same optical properties as glass. And one advantage, we can offer them in different surfaces, so different coatings and so on. So we have optimized them for 2D cell culture, and it's also possible to do 3D cell culture. So let's talk a bit more about that. So unless with glass, we can offer polymer cover slip first and foremost in tissue culture treated. So here on the right side, tissue culture treated means that the naturally hydrophobic polymer was made hydrophilic. And most adherent cell types, not all, but many, prefer hydrophilic surfaces and attach to them without any coating necessary. For example, your cell culture flasks, they're usually tissue culture treated as a reference. We also have many of our tools protein coated, like with collagen 4 or 1. Then we are going to dive more and more into patternings. As you can see, we like our micro in front of everything besides the EB. So our micro patterning is something we are exploring and definitely will put on more focus in the future. But today I would like to focus on BioNerd. So here on the far right side. BioNerd is our fancy name for ultra low attachment surfaces. I think everyone knows what an ultra low attachment surface is, and therefore this is suitable for 3D cell culture. So let's have a closer look on that. BioNerd is short for biologically inert, so no biological reaction. And it's a polyol hydrogel coating that we put on top of our polymer cover slip. So it's a passivated surface on which nothing attaches. Not within a day, not within a week, not within 2 weeks. So it's even actually a no attachment surface, not low, but no attachment. And here's an example compared to tissue culture treated surface where the UVEC cells in this case attach nicely since they're adherent cells. On BioNerd, they cannot attach to anything, and so they're warm spheroids. And we have them in different labware geometries, for example, 8-well, 4-well, a dish. But today I would like to focus on this unique design, this channel slide. We call it microslide spheroid perfusion. And as the name implies, it is for perfusion of spheroids. It comes with 3 channels, and each channel has 7 wells. And each well looks like this in a cross section. So we have a channel on top, and at the bottom is a so-called niche. We call it niche. It's a smaller well. And the whole well, not only the bottom, the whole well is covered with BioNerd. So nothing attaches. And this light can be connected to pump systems. That's why it's a channel format. We use the lure system, meaning the slide itself has female lure adapters, and you can connect male adapters to connect them to tube and then in the end to a pump. For example, our ibidi pump system, which looks like this. Just a few words on it. It's an air pressure pump, and it uses, or it recircles the medium. So it's a closed system. And you can put one part, this here, into an incubator. This part holds the medium and the slide. And with this combination, having a closed system that can be put in incubator, you can do long-term flow applications. So really for days or even weeks. And the slide itself works as follows. You can see preformed spheroids. And this one is open. So if you have to close the channels with a cover slip that we provide. So we have sticky foil on top. And then after you place the spheroid, you can close the channel with an extra cover slip. And as soon as it is closed, you can connect it to a pump system and then apply a very low flow to continuously exchange the medium. That's the whole purpose of perfusion spheroid. And the flow profile looks like this. You see that the medium's only passing in the upper area of the well, and that is on purpose because we don't want to disturb the spheroid inside of the slide. The spheroid is sitting at the bottom and gets the nutrition, the fresh nutrition from the medium exchange only via diffusion. So it's not disturbed, it stays free floating, and you can do long-term cultivation like this without using a pipette and exchange the medium every second day. We actually had a look on the growth of a spheroid under these conditions. This is definitely a long-term experiment done with our ibidi pump system in combination with the slide. And we cultivated fibroblast spheroids for 14 days without flow, meaning medium exchange with a pipette every second day, and underflow, so very low medium flow for 14 days. And you can see here in dark blue, this is the size or the diameter size of the spheroid underflow. And it's much larger compared to the one in light blue that was statically cultivated. So there is a difference, and the nutrition supply seems to have this or make the difference. But of course, the slide, since we talked about imaging facilities, is also for imaging. So this is a nice way to keep spheroids free floating and do, for example, confocal imaging. So this is a confocal stack. Again, it's a fibroblast spheroid. The spheroid has been cleared before, of course, otherwise it's a bit difficult to see through. And very, very beautiful, in my opinion. And if you're looking for a clearing protocol, we have this on our website. So if you're looking for something like this, just search user protocol 11, and there you get the whole clearing protocol inside of this particular slide. And last but not least, for this slide, I wanted to mention you cannot only use preformed spheroid, you can also have spheroids formed in this slide by seeding adherent cells, not attaching a pump, but let the cells settle down. And since it's bio-inert coated, they cannot attach to anything and then will accumulate at the bottom and form a spheroid. And with a scalpel, the cover slip that is glued onto or stuck onto the channels to close them can also be removed with a scalpel. So you can even retrieve the spheroids afterwards, just as an information. Okay, so I introduced you to the pump system. Hopefully I made it clear that you can perfuse spheroids with it and long-term for very long time. But you can do a lot of different kind of assays with a pump system. You can do rolling adhesions, so many 2D cell experiments as well, study barrier function, cell polarity assays, transport studies. So lots of possibilities. If you're interested in those possibilities, we have a recorded webinar at ibidi.com. Just Google book webinar and cell culture underflow, and you find that. But of course, to perform this particular assay, as mentioned here, barrier function, co-culture, and so on, you also need the particular slide for that. So I only focused on one today. This one, microsite sphere perfusion, but we have more solution for 3D cell culture. This one here, for example, is for shear stress studies on top of a gel to make it more physiological. Here you can put spheroids or tissue samples in and perfuse them. And of course, we have solutions for 3D applications, so for adding defined shear stresses to a monolayer of cells. And with that, I just wanted to mention, if anyone caught your eye, go to our website, ibidi.com, and get a free sample to test what cover slip material works best for you, what surface, what geometry, and what is overall the best solution for your experimental plans. And with that, I thank you very much for listening, for your attention, and we are always reachable via chat on our website or via email. Thank you very much.
Great, thank you very much, Peggy. I always enjoy looking at your presentation. You mentioned the webinars, and I know that you also have trainings. I don't know whether that's possible. Maybe you can leave a link in the chat later on for for the fans. It's possible.
Definitely.
Just to let you, yeah, great. Just to let you know that you can leave your questions in the Q&A as well as the chat. We have one question. Here, are the ibidi slides suitable for, I'm trying to pronounce good, InQ site, which is probably
by
microscope.
Sartorius.
Yeah.
In principle, the slide size is standard size, so 25 by 75. So you can definitely put it in, but we are not inside in the lab software. Sorry, we're not included in the software. So you have to manually get the positions into InQSite, and then you can use them definitely. But it's not pre-programmed, if that was a question, unfortunately.
Yeah, great. Please leave your questions in the chat. I'm going to ask a few questions myself. So if, for example, I'm a biologist, I look at your webinars, but still have questions, how can I get help?
The chat, definitely. It's not a robot. We often ask if our chat is a robot, but no, those are my colleagues from the in-house team, and they're really happy to chat, and we're always trying to help. We have many distribution partners in many countries, so they are also very helpful, and just send an email or a phone, but I think it's quite unpopular nowadays to pick up the phone.
Yeah, indeed.
Everyone prefers chat or email. So yeah, reach out anytime. And since you mentioned it, we're also having a course on 3D cell cultivation. So where we go into more detail on this slide and more of our other 3D cultivation slides. It's happening on the 6th or the 7th of March. I definitely put the link into the chat. Thank you for this opportunity.
Great, yeah. So it's funny that it coincides with our masterclass, but
yeah, it was the same last year, I
fine.
think. Oh, right, yes, it's also on the 6th, yeah. But those that didn't get a spot at the masterclass, we offer.
Yeah, indeed. No, that's okay. I had a question about the spheroids. So you mentioned creating a spheroids in the chip in different formats of the chambers. What if I want to control the size of the spheroids? Is there any way, for example, at the time of seeding, or is it always the same size in the chambers?
It should always be the same size, because when you
let
me go back, when you see the same cell number, they will all accumulate here at the bottom. So they should definitely have more or less the same size. And then it depends how you treat them.
Right.
Sorry.
And this is independent of the cell suspension or cell suspension concentration.
They will settle equally. So you close the channel, and then you see the cell suspension. But at channels usually have the tendency to have the same distribution of the cells over the whole area. So if you seed as much as possible and have something go out on the other side, you will make sure that it's more or less the same number of cells per well.
Yeah.
Probably not 100%, but you will be good.
Yeah.
It should definitely be the same size. Yeah.
And you probably showed it in your presentation. What was the size of the spheroid in these
not the spheroid, but I can show you the size of the well. So this well here at the bottom is 800 micrometer in diameter.
Yeah. So we get an idea of the size of the spheroid.
Yeah, exactly.
That's clear. Thank you very much for the nice presentation. I don't see any questions, but if there is questions still for Peggy, please leave them in the Q&A. We'll try to make time at the end to answer the questions.
Perfect.
Okay,
Thanks.
great. Thank you very much, Peggy. I would like to invite Elena from Microfluidic ChipShop to share her presentation.
Yeah, thank you very much.
Thank you, Elena, for being here as well.
Yeah, so hello, my name is Elena Müller and I'm from Microfluidic ChipShop. We are a company in Jena, so in Germany, and we are producing microfluidic chips, a lot of different varieties of chips. So our basic mission is to shrink the biological and chemical laboratory to bring lab-on-a-chip systems into the daily laboratory life at a reasonable cost. So we mainly divide our work into these 4 different parts. We're doing a lot of customer-specific development and manufacturing. We also offer complete systems, which is an instrument and a cartridge all-in-one solution to have a lab-on-a-chip device. We are also doing a lot of collaborative R&D projects with universities and other research partners. But we also have this huge catalog. It's 350 pages, the newest one, where we offer a lot of off-the-shelf products, chips, but also accessories, different pump systems, and basically everything you need to get started with organ on a chip, or basically cell culture on a chip projects. So our chips that we offer have different features. So our basic interface type is the standardized mini lure interface, which is a smaller version of the lure. Therefore, more interfaces will fit on a microscope slide compared to the lure interface. We also offer the appropriate connectors for that, so you can connect your chip easily to pumping system and tubings and so on. The standard chip format we have is a culture slide, so microscopic format, which you can basically use on every common microscope and fits to every equipment you have already in the lab. We have different chip materials, meaning different thermoplastic materials, which are injection molded. All these materials show a minimal absorption of small molecules, and depending on the application you want to have, we can give you a hint of which materials are generally good to use. They are all compatible with microscopy, so fluorescence microscopy with a very low autofluorescence values. For adherent cell culture, we also offer an hydrophilization treatment for better adherence of the cells to the chip material. We also have some functional elements, or can have some functional elements in our chips, like pillars or also membranes to make co-culture experiments. As I already said, our chips can be easily connected to pumping systems. Doesn't matter which pump, basically peristaltic pumps, syringe pumps, or pressure-driven pumps work all the same. And that already I mentioned. So the readout can be a microscopy readout. So generally, I want to say that our chips differ in their features. So we have here, for example, some simple chamber chips where you can just culture monolayer cells and stain them. We have some more complex chips also in the slide format, where you can culture, for example, 2 different types of cells and study the interaction of the cells, like simple organ-to-organ interaction studies. Then we have these membrane chips, which have an, you can see this here in the scheme, membrane with a specific pore size, and you can, for example, study a barrier functions, a blood-brain barrier, for example, and here in this case, liver simulation. And yeah, we also have more complex chips. This is a chip in a tighter plate format, also with electrodes. So this comes more into the customized development for, yeah, some specific applications you might have. So I basically want to show you, I think, 3 or 4 different types of chips that might be of interest for you for cell culture and organ-on-a-chip applications. I already mentioned these cross-flow membrane chips. You can see here in this picture the membrane in blue. Then we have a lower compartment in green and the upper compartment in red. The membrane we integrate in these chips can have different pore sizes, different pore densities, and also thicknesses, which all lead to different light transmittance of the different membranes. So depending on your application, whether you want to have a barrier study or you want to study, for example, transmigration of the cells, you might want to choose the pore size accordingly to the experiment. We also have, from this type of chip, a more complex chip. You can see here. So this has 4 chambers that are all these transwell-like chambers. And this chip is, was designed to study the liver and kidney functions and interconnections between these 2 organs. You can all seed these chambers separately, so they can have different cell types, and then study small molecule signaling. Next type of chip we call the interaction chips. These are basically chamber chips where you can seed your cells as a monolayer, but they have interconnected chambers. So for example, this chip here has 2 connected chambers, which can be seeded with different cell types, and then you can study the interaction. This chip has this special channel that is connected to the main chamber via pillars. So you can check some signaling molecule distribution and other experiments. This chip is another option when you want to study the connection of more than 2 different cell types. You have here the opportunity to have 3 chambers. Next, we have the also the option to have, for example, matricel, so extracellular matrix simulation in these chips. This chip is designed with 3 columns. And the main column, you can seed your cells in the extracellular matrix. And the outer 2 channels can be used to supplement nutrients or use them for migratory cell assays. Another chip family I want to shortly show you is the suspension cell chip family. So this comes with a special handling frame also. And this family is designed for the cultivation of non-adherent cell types and also cell clusters like spheroids. So with microfluidic chips in a flow system, you always have the problem that you will just flush your cells out. And these chips are designed so that this doesn't happen. So here you can see that this chamber is lower than the flow channels. So your cells will stay inside this chamber and will not be flushed out. These chips can be then also used for microscopic analysis. And this handling frame that we also designed here in-house allows for a very minimal working distance between the objective and the chip. So you can do high resolution imaging with these chips. And also 4 chips fit into this handling frame, so you can do multiple of analysis. Next, I want to show you a chip that can be used for spheroid cultivation specifically. It goes a bit in the direction what ibidi showed before. So you can cultivate spheroids or organoids. You can either use preformed spheroids and seed them in there, or you can generate the spheroids directly on the chip. As you can see here, these chips will be collected in a kind of a catcher structure. So you can just see the solution with all the spheroids in there, and they will be caught separately in these catcher structures and will remain there. You can apply a constant flow to exchange nutrients or have some drug treatments to the spheroids, and they still remain inside these structures. And of course, you can also do fluorescence staining of the cells. Yeah, that's I was quite fast, but that's from my side. So as you could see, we have a lot of different chip versions. Basically, I think fitting all of your investigations you want to make. So I will send the link to our catalog also in the chat, so you can just have a look yourself or also ask questions. And in general, this email address here is the best to ask us any questions you might have. So thank you.
Great. Thank you, Elena, for the presentation. I can personally spend hours in your catalog searching for stuff. It's quite extensive and also quite complex. Thanks. So if you can just put a link to the catalog, that would be very helpful for people. So if I, before asking the questions, if there's any question, please leave them in the Q&A or chat, then I will announce them. And if you have any questions and you want to say it yourself, please raise your hand and then I will unmute you. So my question was mostly about use cases. So you have a very complete catalog, but if I want to know more about previous experience, your use cases in the past, or your partners have tested your chips, how can I access those data?
We have in the catalog for at least most of the cell culture chips some papers stated there that use the chips. We also have some application notes on our website to show how these chips work, because I know it's much. I just showed some basic pictures, and probably some of you don't even know how to handle those chips. So we have
and
these
of course it was
applications.
short. Talk about everything.
These applications note with pictures, how to use, how to get started, how to connect them to pumps and so on. And if you have any special questions, you can just write an email and we will answer that.
Okay, great. And of course, if they join the masterclass in London, they can directly ask the questions from you and then have a nice chat, because there is enough time for everybody to discuss.
Yeah, and also there you will get hands-on experience. So we will bring all the chips I showed you and more, so everyone can have their try on filling them and really seeing them in action.
Great. It will be a nice experience, in my opinion. Okay, great. Thank you very much. I don't see any questions. Maybe something. Yeah, that's your link. Thank you very much, Elina, for your presentation. I would like to invite the next speakers, Naina and Laura from Netri, to share your presentation. Thanks for joining us, both of you.
Hi. Okay.
Hi.
Hi, Hussein. Thank you for introducing us and inviting us to your masterclass. So I'm Naina Salman and I'm joined by Laura Rijal. We will present Netri and the solutions that we offer. So at Netri, we say that let's innovate, screen, and then predict. So first off, who we are a France-based medtech company which believes in digitizing the human biology, and we've been founded in 2018, having 200 meter squares of production facilities. We have about 70 plus clients all over the world with 12 patents to our name. And the company, at the heart of Netri, we have 30 people consisting of neuroscientists, data scientists, electrophysiologists that work for the R&D part of Netri. So the first of what Netri provides is a high throughput and interoperable solutions for organ-on-chips. In most drug compound, the problem with the research transportation from animal model to human model is difficult because most of the molecules do not pass through the first phase of approval. At Netri, we aim at reducing the time and the quantity of experiment needed for approval by mimicking what a normal human anatomy looks like. So what you see on the left is what a standard mixed cell type well looks like, in which you have co-culture, but there is no compartmentalization. Whereas in a real anatomy, there is compartmentalization. For example, a sensory cell that has a cell body lying in the spinal cord would have its axon processes extending up to the tip of a finger, for example. We mimic this anatomy by producing what we have, what you see here, that is a dual link that consists of 3 channels, one on the right, one on the middle, and one on the left. And these channels are connected by microchannels, which allow a compartmentalized manner for the cells to be separated, yet allowing their processes to make connections. What you see on right is our individual chip that's called a dual link. And as I said earlier, so you have a channel 1, a middle channel, which serves as a barrier between these 2 channels and ensures that there is fluidic isolation between channel 1 and channel 3. The Netri dual link chip comes as a production unit that is a quarter bento having 4 data points, and that is plugged into this black frame, which is called a neo bento, allowing you to have 16 data points on one chip at a time. The main advantage of this neo bento, as you can see, is that it is SPS compatible format. As a result, it is compatible to be used with the regular lab equipment, and it has the same configuration as that of a 96 well plate. So moving on, what I said earlier, that with this SPS alignment on a 96 well plate allows the Netri neo bento to be used for a high throughput screening. it is compatible with most of the liquid handling equipment that we have in the lab, and also most of the imaging equipment that are usually used in the lab. Moreover, you have the flexibility to optimize with this your experimental plan, and as such, you do not need any upfront equipment to start your project or your experiments. Moreover, we also have designed in this new bento a very excellent humidification system that allows the new bento to be well humidified even in an incubator for up to 3 to 4 days. So as I said, it's like a standalone device that does not need many equipment to work with. So moving on to the cell types that we have and what are the protocols that we have worked at Netri. We have currently 6 human iPSC cells that we have validated in our devices. We have proper protocols, cell culture technique protocols, and maintenance protocols for these cell types. We also work in partnership with some cell providers, some shown here. With them, you can have all the support from us on the culturing and co-culturing of these cells from these providers. Although it is also, we also help with other cell types and even in-house surgery cells if you plan to go ahead with that. We have also these protocols for 2 human primary cells, the keratinocyte and endothelial cells. Plus, we have the possibility to have the cultures for other cell types, muscles, liver, kidney, etc. as well. So far, what we have discussed is the compartmentalized capability of the Netri chips that allows you to co-culture 2 different cell types and compartmentalize it. Netri has taken this ability to the next level and used this compartmentalization and combined it with electrophysiology recording in partnership with the Axion Biosystems. So I'll just play a small video how it has done. So this is the chip that we have and the plate from the Axion Biosystem. This PDMS chip has been combined to the Axion plate, and that goes into the Maestro, and that allows you to do the electrophysiological recording in a compartmentalized manner. Moreover, you will have the metrics at the end in each compartment thanks to the electrodes that are present under the channels and the microchannels. This capability is also high throughput. And so in the slide, what you see is what our neurofluidic devices look like. On the top, what you have are the simple microfluidic devices coming with in a light version and a full version. So what you have in a light version are 8 data points. And since, as I mentioned earlier, it is SPS compatible, to fill up the space for a quarter bento, we have the plugs. And the same format is also available in the multi-electrode array range, that is the MEA Edge version and MEA Pro version, which are compatible with the Axion Biosystem Maestro Edge and Pro respectively. So what is the main benefit of this compartmentalization and MEA recording that Netri offers? So as I said earlier, that in the Netri devices, we have compartmentalized a microenvironment. As a result, you can have different cell types in different channels. So if you look closely on the right, what you have is what we call a channel 1, and on the left, we have the channel 3. And in between, we have a channel that is the channel 2 that ensures almost 100% fluidic isolations between these 2 channels. So as a result, you can have different cell type on each side, and each side has specific electrode map that is representative of each channel. You also have these electrodes in the microchannels, and these the the recordings from these electrodes are presented in conjunction with the access navigator from Axion Biosystem by our software, which I will explain in a while. So you have the ability to isolate the neurites. So you could have somas on one side and extensions that are going into the microchannels and make synapses to neurons or other cell types on the corresponding channel. This allows you to have study the effect of drugs on specific population while keeping physiological connections and fluidic isolation at the same time. So moving on, so the applications that we have at Netri, at the forefront, we have the neurological disorders that we have the applications in-house. We have more information about these on our website as well. So it's about Alzheimer's disease, and you can have more information on the application or on our website. These applications also extend to the oncology part of it and the peripheral nervous system disorders like pain and pain injury models. We are also in the process of replicating this organ-on-chip in the guts with the involvement of immunology. And we have a major application of the neurocosmetics in which you can have neurons and keratinocytes co-cultured and then recorded electrophysiologically in an isolated environment, depending on the compounds that you use. So one of the biggest advantage of the Netri chips, as I said earlier, was that it is SPS format compatible. So that allows it to be compatible with standard readouts that we have. So if you have, so the readouts that you can do with our chips is that you can do lysis cell analysis for example, ELISA, Western blots, and mass spectroscopy, or you could do the imaging with the live staining, calcium imaging, and immunofluorescence. The chips are made with PDMS, which is an optically transparent material, and our chips come with a bottom cover that can be used for regular day-to-day microscopy, but for high-resolution microscopy, you remove that, and it is also compatible with high-resolution microscopic, most of the equipment that is in the lab. And in the middle, what you see is the main readout that I just mentioned, that is the electrophysiology, that you can have the multi-electrode array readouts in a compartmentalized manner. And that will give you what we have, will be presented like raster plots, and that will be area and channel and per microchannel specific metrics readouts. So we have this capability, the compartmentalization with MEA is also high throughput, and overall the solutions that Netri provides is in the not only just consumables like equipments, the Microfluidic devices, what I showed you, and the different cultures that we provide, but we also have softwares which include the Microfluidic digital part of the Netri, which we are working on. And this part works in hand in hand with the Access Navigator from Axion Biosystem that allows you to extract relevant data, which is in real time, and that is presented in a highly sortable manner. So you have that in like a report plus like a plot that shows what are in real time the effect of the MEA recording. We also provide servicing and services and licensing. That is the R&D FTE, that is the full-time equivalent, is where we can offer to do the research and development according to the market needs. And we can also do the screening of different compounds, again, depending on the needs of the users. And we also believe that we can co-develop models and different R&D in hand with users and companies. So this is exactly what I said earlier. that we at Netri, we believe that the solutions are in your laboratories. You can use our applications, your MEA devices, the neurofluidic devices, and we'll support you throughout. Or we can do it in our laboratories with the innovative skin models. For example, the Alzheimer's models and the Parkinson disease model and so forth. And then compare that to our digital library. And eventually what we aim at doing is to do together a drug development partnership in which the disease modeling and digital libraries will be based on our compounds, on your compounds, and that will be compared to our digital library. So what we mean when we say that we are talking about digital libraries, and what exactly we mean by that. So what Netri offers is a unique take on organs on chip. And within this MEA capability, we can have a drug or a cream that can be tested in our chips. And from that, we can know, from the response that we get at the neuronal side, we can know if a certain drug or a compound has a validated formula, or the formula is not validated, for example. We can feed this information into, save that information, and that can be relevant. How it is relevant? That we do not see skin just as an organ. We see just skin. It's actually just, it could be any organ. And we do not see neurons as neurons, but they are actually biodigital sensors, which allows us to digitize the human biology. So what you see here is an example of the skin cells and the neurons on the other side, but it could be any other organ that you have, and the digital signature from this organ recorded by a neuron will feed into what we call the digital library. So this is a schema of what a digital library is going to look like. So we plan to have 3. It's the same Neobento as I said earlier. So what we plan to have, that we will have 3 reference compound, for example, in 3 of the quarter bentos, and you will have 1 target compound. And the MEA recording, the electrophysiology recording from these 4 compounds, 3 reference and 1 target, will get processed in the network through Axion Maestro, and they will be fed into the AI digitals. They will have an AI digital signature. And with this library, we will be able to predict the efficacy of the drug in comparison of the match to the reference compound. This will allow us to predict in vivo the target compound without in vitro predicting the efficacy of the target compound without and that will eventually shorten the length of the drug approvals. So to sum it up, what Netri offers is a unique selling proposition. It is high throughput and interoperable solution. It is compartmentalized. It is a standalone device that does not need any pump or any specific lab equipment. And the readouts, and for most manipulations, is compatible with most of the lab equipment. So you can literally begin with just 1 chip in hand, your experiment in your lab to begin with. And when you take it to the next level, we believe that neurons are the biodigital sensors, and all the cell, all the organs can be used in place of a skin. The signature from this can go into the digital library, and we hope to reduce the timeline between drug trial and approval by studying the efficacy and toxicity of different compounds. So that's from my side. Thank you very much.
Thank you very much, Naina and Laura, for the nice presentation. It's nice to see how scalable your model can be. So we are open for questions. Please leave your questions in the chat or Q&A, and then I can read your questions. So you said that your solution is pumpless, which is nice. But if I want to create flow in a specific organ, how would you suggest? What are the ways that I can create flow in your chips?
Okay, for the moment, the main strength of Netri lies in the fact that we don't need the pumps. And it is a 96 well plate format chip. If you want to do that, that would come in the form of the well that we are working on. That is like a 3D version of the Netri devices. And we hope to have that in the future. But for the time being, the PDMS itself allows gas exchange, it allows humidification, and with the specific design, you don't need that. To create the shear, what I understand is that we are working on another 3D device, that is the duplex well, which will be available mid-2024, in which you will have the possibility to do that if needed. Even then, it will be pumpless if you want it to be like that.
Yeah, in other words, the media that is available to the cells is already enough in your system.
Yes, and we do otherwise recommend regular changes of the medium in 2 or 3 days, and that works absolutely fine. Very good.
Yep. Okay. I look forward actually to seeing your new solutions for creating shear stress on the cells.
Thank you.
And then maybe a bit more to the practical part. I'm sure that you're gonna teach a lot to the participants, but maybe a simple question: how do you inject cells into the chips? Maybe for the participants who are new to organ chip.
Okay. So what if I share my screen again? So as I said earlier, that our chip is basically there are 3 parts of the chip. So there is like the channel 1 and channel 2, and so what we have is here. So these channels, all of them, this is a dual link. We have another architecture that is a trial link, in which this middle channel is quite broader. It is as broad as channel 1 and channel 3. In that architecture, you can actually culture seed all these channels. But in a dual link, what we do is we culture it from here, and we have a recently optimized cell seeding protocol that we have worked on. And in this protocol, we just require 3 microliter of your cell solution to seed your channel 1, and you have the same cell concentration, cell density as it was seen with other microfluidic devices, which require about 25 to 30 microliter of your cell seeding solution. So yes, we help with that as well, how to, for people who are first-time users of the microfluidic devices, we help them get trained on that as well.
Yeah, that's clear. And do you have also problems with non-specific binding in the system?
Binding
of molecules?
Binding. Attachment of the cells.
So I mostly specifically refer to non-specific binding of molecules, for example, drugs in the system.
No, for most part, PDMS is it does not allow that, and along with the coating, it's not the problem that we see. And as I said earlier, that there is a fluidic wall between the two. At least if there is a compound here, it will not seep over onto the other side. So there will be fluidic isolation on one side.
Right, right. I also very much like, in addition to microscopy, you have this electrophysiology readout in such a scale, which is really impressive, I think. And yeah, that was my questions. Thank you very much, Naina and Lara, for a nice presentation. I look forward to meeting you in person, Naina,
Yes.
in London.
We look forward.
And we definitively will come with our devices, with MEA, and also with a proof of concept with the 3D microfluidic devices, right?
Yes, exactly.
That's perfect. That's perfect. Thank you very much.
Thank you very much.
And I would like to invite Dr. Lisa Hulting from InSphero. Thank you, Lisa, for being here and presenting InSphero today.
Yeah, so hello from my side, and yeah, welcome to my part. My name is Lisa. I'm a biologist by training. I have a PhD in stem cell technology and in vitro disease modeling. I joined InSphero almost 5 years ago to support the development of 3D in vitro models for safety assessment and drug efficacy testing. Before I will now talk about our advanced Acura organ-on-a-chip platforms, some words about InSphero. InSphero is located in Zurich, Switzerland. We have currently over 75 employees, and over the last 14 years, we have established a global customer base, and meanwhile, we have the capability to produce every month over 30,000 3D in vitro models, which makes us to the market leader in 3D assays. So to sum up, what we do at InSphero is that we combine tissue and disease modeling with microplate engineering and endpoint technology development. So let's start at the beginning, and at the beginning, everything starts with the formation of our 3D spheroids. So you can see a time lapse how the cells aggregate to 3D in vitro models, and we call our models microtissues. So we at InSphero have different types of spheroids. So we have 3D pancreatic islets microtissue to study diabetic research. Then, of course, we have liver microtissues on the one hand for predictive and investigative toxicology, and we have 3D liver models for disease or liver disease like MAFLD and MASH for drug efficacy testing. And of course, we have a couple of tumor models for immune oncology related questions. So I will now focus not on a particular model, but I want to focus on how we at InSphero study tissue crosstalk. So the approach that we have is that we want to add a new level of complexity to our already existing 3D models. So we want to add physiological flow conditions. We want to add immune cells, or we want to study tissue interaction. So this is what we want on the one hand side, and on the other hand side, we want that the platforms that we are developed, that they preserve our scalability, which means that all our platforms are compatible with automation and the standard lab equipment. Then we also want to preserve the robust handling, which means that for all our platforms, we want to keep the operational complexity very low so that we prevent microtissue loss. And of course, we want to have a platforms that are compatible with our already established endpoints like biochemical assays, high content imaging, or transcriptomics, so that we don't need to invent everything from scratch again. And as you may, or if you as you could imagine, that this is not a development that is taken place overnight. And this is nicely reflected here in this timeline. So the first initial trials have been taken already 13 years ago. At this time, our VP, Olivier Frey, was a postdoc at the ETH. And then it took a couple of years over the first functional prototypes to an automation and industry compatible model. Model in this case, our chip version. This is our Cura Flow version. And we further developed the chip in the last years, and we came up with 3 new platforms. It's the Twin, the Flow 384, and the Immune Flow chip. And I will now talk a little bit in more detail about the different platforms. Due to time reasons, I will focus more on the features of the plate, so that you can maybe get some impression and think about how the platforms could be beneficial for your own research. Because the platforms are actually tissue independent, so it only has to be a scaffold-free model, and it needs to be pipetable. So all our platforms follow the same principle that we call an off-chip production and on-chip integration, which means that all our models are produced in the 96 or 384 plates according to the standard protocols. Then they are QC'd to be sure that only functional microtissues are transferred and are combined in the chip. So here again, the workflow is shown. So we have the production of the individual microtissue models. And by combining them at a later time point in the chip, it's also possible to combine microtissues that needs different time of differentiation or generation. Then we have, of course, protocols developed to do a manual transfer, but we also have semi-automated and automated transfers to transfer the microtissues from the production plate into the advanced platforms. Then our long-term culturing and treatment is in a pump-free and tube-free system. So we induce our flow based on gravity forces. So we have the different platforms as a plate or in a frame that we can put on a tilting device, which has the size of a shoe box, which easily fits into the incubator. And by programming the tilting angle, the speed, and also the tilting cycle, we can modify the flow. And then, of course, we can combine that with our already established readouts. So let's talk about the Cura Twin platform. This is our simplest organ platform that you can see here. That's not. Yeah, so that you can see here. The body is our standard 384 platform, which means it has an ultra low adhesion coating, and then we also have this feature of the shoe exchange ledge, which makes it possible to perform a full medium exchange without microtissue loss. But the unique thing of our twin plate is that the body has a COP bottom, which is a specific design, and this design allows the connection between 2 adjusted wells. So as you can see here in the cross section, that always 2 wells are connected via this thin channel. The plate was actually developed in cooperation with Roche, because Roche requested a platform where they can test for immune immune-related drug-induced liver toxicity by bispecific antibodies. So they wanted to have a platform where they can measure on-target effects, so the effect of the bispecific antibodies on the tumor, so measuring tumor shrinking, and on the other hand, see if there's also some off-target toxicity, for example, liver toxicity. And this is how it looks like in our plate. So this is the image of one twin well. On the one hand, here shown in green, you have our 3D liver microtissues. In red, you have 3D tumor tissues, and they are surrounded by immune cells shown in yellow. And what you see, of course, it's a very simple organ system, but it has the advantage that it's highly scalable. So on one plate, you have up to 192 conditions, which makes which allows you to test a lot of different conditions with a lot of replicates. And now some words about the Acura Flow 384 chip. So this is actually the direct development of the Acura Flow chip. So it's the transformation from a chip to a plate design to even more improve the scalability. So again, the body is a 384 plate, and then on the bottom we have a channel structure. So here in the cross section, you can see that. So what we have is we have 12 individual compartments that are connected via a thin channel. And then on the top and on the bottom, we have a medium reservoir. And by tilting back and forth, the liquid flows from the upper reservoir along the channel, interconnecting the individual compartments to the lower reservoir, vice versa. And the advantage is that this is a top open top approach. So you can, as you can see here, you can add and of course also remove again the microtissues. And due to the thin polystyrene bottom, it's also image compatible. And of course, you can always sample the liquid. If you now ask the question, okay, why 12 compartments? I don't have 12 different organs in mind that I want to combine. I can tell you that you don't need 12 different organs. You also can add 12 single organs if, for example, you want to increase the cell-liquid ratio, if you are, for example, interested in some rare metabolite that you want to measure. Or you can combine two types of organs and then play a little bit around with the ratio. So for example, we have co-cultured islet and liver microtissues under different metabolic states and measure insulin secretion on the islet side, and we check for the lipid content and glycogen content on the liver side. And now I will also tell you a little bit about our Acura Immune Flow chip. So the Immune Flow chip is actually the chip that combines flow, different tissue-tissue crosstalk, and immune cell interaction or tissue suspension cell interaction. And if you now ask the question why you have an additional chip, why don't you just simply add your suspension cells to the Acura Flow or Acura 384 plate? The reason is that we realized that while adding our suspension cells to the Acura Flow chip, the cells quickly, or the suspension cells quickly sediment, then they can clog the channel or they somehow accumulate in the reservoir and they are not really constantly in suspension. So that's the reason why we came up with the Immune Flow chip, which we designed with the ETH group. So from a straight line that we had in our previous version, we designed a meander-like structure. And with that, we can constantly keep the cell in suspension while tilting the chip. And this is how the chip looks like. So you have here 7 different compartments to culture 7, for example, liver microtissues, or you can also culture different organs. And then you can add the cells in suspension. And here, according to the streamlines, you see how the cells are flowing through the chip, and that there, our cell interaction chamber, there's enough connection or possibility that the immune cells or cells in suspension can crosstalk to the solid tissues. This is one example. Together with the Erasmus Medical Center, we have a project where we co-culture liver microtissues with prostate cancer cells. And what you can see is that over time, the prostate cancer cells start to attach to the liver microtissues. And so this could be a potential application to test for metastatic potential of different cancer cell lines. Also loading different tissues, so to test if there is a preference for a specific organ to attach and invade. So putting this all now together, we have different platforms, and depending on your question, the application and requirements, you can choose between the different platforms. And I will bring all of them to the masterclass in London, and then you have the chance to work a little bit with them, maybe perform some medium exchange, to get an impression about the scalability and the complexity of the individual system. And with that, I would like to thank you.
Thank you very much, Lisa, for the nice presentation. I'm also looking forward to seeing you in London and seeing the new chips. I haven't seen the immune chip yet, so that would be cool to see.
Great.
Okay, so before starting the questions, there are some questions from audience, and I would like to ask the speakers, if possible, leave your email addresses in the chat, and if possible, again, your LinkedIn profile so they can follow you. This is a request for all the speakers, so you can put your information in the chat. We can also have questions from Lisa. I'm going to start questions. So I'm a researcher and would like to buy or try your chips. What are the ways I can actually do that? Should I buy them? Can I also test them? Is there any way?
So you can just start with contacting us, and then we can discuss the project. So of course you can buy all the different products, and we offer them with also some starter introduction training. So either online, or you can come also to us to get a hands-on training. And so from time to time we have some so of course we have academic prizes, and from time to time there's also, if this is a special interesting application and topic, we offer there some special academic.
Great. So all the chips are available commercially, they can buy it online. Okay, that's clear. So especially for Acura Flow 384, you had 12 chambers. I know this question has come in the past. Maybe you can elaborate on this, that this is you can also use it for organ-organ interaction, right? The question was, how do you determine the number of the tissues? You know, the number of each tissue, but also the number of, for example, liver tumor model, the ratio between the liver tissues to the tumor tissues.
Yeah. So I mean, just one example with the liver and the islets. So we checked how it is in the body and somehow scale down the ratio. And on the other hand, or in parallel, we also tested if this number is sufficient. So if, for example, the number of islets enough to secrete enough amounts of insulin that it has an effect on the liver. So usually then you also do some ratio tests to figure out if the ratios also make sense from the functional point of view.
Yeah, that's clear. Thank you very much. Now, there's a question from all the speakers. Maybe I can ask it from all the speakers. Anybody who has suggestions can answer. And this question is related to young researchers who are graduating from a 3D bioprinting program or microfluidic program. One of the audience wants to know what kind of skills they need to develop to work on an organ-on-chip company or microfluidic related company. Do you have any suggestions? And this question is targeted at all the speakers. Maybe you can help them a bit.
Yeah, maybe I can start. So I
Sure.
would say, of course, first to have interest in the topic. I think to be motivated and have a great interest in what you are doing. I think it's one of the first requirements. Then, I mean, I'm a biologist. I'm working with a lot of engineers. So it's always good to have a little bit of understanding from my side, from the engineer part. But I think this is, yeah.
Okay, that's clear. Any other suggestions from other speakers?
Well, it depends on which department you would like to go in. I mean, if I just guess, because everyone wants to work in R&D, then I agree with Lisa. Those are the requirements, but there's also marketing, there's sales, there's product management, there's so many different opportunities. And then more the personal, your character will matter most, especially in sales, and your social skills. Of course, it's good to have all the background to be able to explain it to potential customers. And for marketing, also technical background is important, but then you must be good in writing and expressing and explaining things. It really depends where you want to be in a company, I'd say.
Sure.
Yeah.
Maybe I can just confirm what you said. The most important part for me was, when you're a PhD student or postdoc, you have one project or you're helping with some other projects, but in the company, you may be multitasking, communicating in different projects. You have to maybe sometimes not related to the science. You have to do, as you said, sales as well. So, and sometimes, it comes on the way when you start working in the industry.
It's also true.
Yeah. Okay. I would like to thank all the speakers. Thank you very much for your kind presentations. The last remarks, I would like to invite Stephanie also join us. So we are going to have a masterclass in London, 7 and 8 March. You're getting the contact information, by the way, in the chat. If you have any questions about the masterclass from any of the company members or from us, please send us an email and we can explain things to you further. There is a room to register for the masterclass, so be quick because we are going to start with the e-learning module, and then that's really the deadline for registering. And I have no addition. Stephanie, do you have anything to add?
No, nothing to add. Just as Hossein said, there's still places open for registration, so do spread the word. And really excited to see everybody there on the 7th to 8th of March. Thank you.
Yeah, so Stephanie, we have a question. How do we apply for the early career researcher discount for the masterclass?
So if you Click the link. Basically, you will find the information on the pricing there, and then you can get in touch with us at Animal Free Research UK to get the discount code.
Yes, I'm putting the link for the masterclass now here. And via this link, you can go to the registration website and there is more information on how to register for the masterclass. Okay, thank you very much. If there's more questions, you have all the email addresses in the chat. Please feel free to send us emails if you have any questions. Thank you all very much and hope to see you soon in London. Have a great day.
Yeah. Thank you.
Have a great
Thank you.
course.
Thank you very much.
Bye.